Hello, everyone, and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar that is organized by the GastroPlus user group. We have two speakers for today's webinar. The first one is Grace Fraczkiewicz, I believe that, Fraczkiewicz from Simulations Plus. She will give us an overview on experience with regulatory submissions using GastroPlus mechanistic absorption and the PK modeling. The second speaker is Dr. Da-Bing Chen from Boehringer Ingelheim. Da-Bing also is my colleague at the Ridgefield, Connecticut site. He is going to share with us a case study on using GastroPlus simulation to justify drug particle size specifications for an extended release formulation in support of NDA filing. Grace is currently a team leader of the simulation studies team at Simulations Plus, where she led a group of scientists providing mechanistic absorption and PBPK modeling consulting services for the pharmaceutical industry. Projects performed by her group encompass all areas of pharmaceutical product development, from early discovery through clinical development. And she has been involved in numerous studies that inform the regulatory decisions. Grace joined Simulations Plus in 2000 as a scientist supporting the development and the validation of new software, such as GastroPlus, Adam Predictor, and the DDD Plus. She has been leading the consulting service team since 2010. She holds a bachelor degree in chemistry and a master in chemical physics from Wrocław University in Poland. Da-Bing is currently a senior principal scientist in material and analytical science of Boehringer Ingelheim. He holds a bachelor degree in pharmacy, master in pharmaceutics from Beijing Medical University, and a PhD in pharmaceutics from University of Minnesota. Da-Bing's research interests include enabling formulation for polywater soluble drugs, salt and co-crystal engineering for improved bioavailability, mechanistic modeling of dissolution and oral absorption to develop in vivo predictive dissolution, and the crystallization and the solid-state characterizations. So this webinar will be recorded and it will be available to the users group members in the future. Following each presentation, and there will be an opportunity to ask questions. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. So this will enable us to unmute your line so that we can hear you. So without further ado, we now begin the webinar presentation with our first speaker, Grace.
Thank you, Sherlyn. So I will talk about our experience with regulatory submissions using GastroPlus PBPK modeling. I will give you first some overview of number and category of regulatory submissions. Then I will go to key points to be addressed to regulators. I will point some differences between the FDA and EMA requirements, and at the end I will give you examples of submission responses from regulatory agencies. Based on the data from the Office of Generic Drugs, there have been 34 submissions related to mechanistic absorption modeling from 2010 to May 2016. 15 of them were related to IR products and 19 of them were related to extended modified release products. Based on the very recent publication, actually in Journal of Pharmaceutical Sciences, published by the FDA researchers, number of NDA submissions containing PBPK analysis to the Office of Clinical Pharmacology between years of 2008 and 2017 was 94. Total intended applications were higher than that, 110, since sometimes one PBPK model was submitted for more than one area of application. As we can see from this table, the highest number of submissions was related to SIP-related DDIs. The second highest number was related to pediatric population predictions, and somewhere in the third place would be the oral absorption related submissions. We obviously don't do regulatory submissions ourselves, but we provide consulting services to the pharmaceutical industry. And our consulting team worked on over 110 projects since 2016. And some of these projects resulted in regulatory submissions. But we often do not know about the outcome unless the project sponsor contacts us asking for help with regulatory response to their submission. And in some cases, we know about the project was strictly intended for the submission. So, however, sometimes we cannot fully share the experience due to the confidentiality reasons. And going back, you can see that the projects pretty much encompassed all the areas of pharmaceutical development. The things that the regulators look for during the submissions is the objective and intended regulatory purpose of the PBPK modeling. They want to have sufficient background information to place this modeling in its context, in the clinical development of the drug and the reason for the submission. They want to see good model validation and the discussion of the data used and assumptions made during the model building. They want to see supportive data and also impact of the assumptions, discussion of the impact of the assumptions on the model and the outcome. Very often they ask for sensitivity analysis, especially on the parameters that were fitted in the model. And there are the requirements when it comes to reporting and the data required for submissions are quite similar between the FDA and EMA, with one major difference, that FDA also requires the model to be submitted, so the model files to be submitted to them, since they have capability of running the model themselves and checking whether all the information stated in the report is correct, and also sometimes do additional analysis themselves while responding to the sponsor. EMA doesn't require model files to be submitted, only report. So now I will go to the first example, which shows EMA regulatory outcome. The example was related to BCS class 4 compound, which was primarily metabolized by CYP3A4 and to some extent by CYP2D6. The compound was also a strong inhibitor of CYP3A4 metabolism, including self-inhibition via this pathway. So it was one of these cases that FDA and EMA and other regulatory agencies have still problems with accepting due to the reasons that I will discuss later. The reason for this submission was that EMA didn't fully believe that the drug was cleared only via metabolism pathways and asked the sponsor to build PBPK model that would explain the compound's disposition and clearance in humans. They also wanted them to use this model to predict the compound's interaction with midazolam, for which in vivo study was already conducted, and the data, in vivo data, were available. So there were The following assumptions were made during the model development and simulations. The model used apparent clearance. Basically, the in vitro Vmax was not representative of in vitro Vmax for CYP3A4 was not representative of in vivo clearance via this pathway. So the multiple dose data for this compound between 50 milligrams and 300 milligrams were used to optimize Vmax for CYP3A4. Also, the in vitro KIs for these compounds were not predictive of in vivo. So since the data were available for multiple dose administration of this compound between day 1 and day 14, these data were used to basically optimize KI in vivo K inactivation, to obtain in vivo K inactivation, and the study with in vivo study with midazolam was used to further optimize the KI for this compound. And yeah, the justification for this was basically that the compound is a strong inhibitor of its and autoinhibitor of its metabolism, and the in vitro metabolism was most likely already affected on day 1 during the measurement of and for that reason the in vitro values were not reliable and not predictive of in vivo processes that we were trying to capture. So the outcome was such that EMA was impressed with the quality of the model. Agency accepted that the compound is primarily cleared via metabolism, and agency asked the client to conduct another clinical DDI study, the compound co-administered basically with another strong CYP3A4 inhibitor to prove that the fitted inhibition parameter is valid. And for this reason, the submission is considered to be partially successful since it changed the EMA's mind when it comes to the routes of compounds clearance. However, the agency still required additional DDI predictions for this compound, DDI studies for this compound to be conducted. The second example of submission to both agencies, FDA and EMA, is related to BCS class 2 compound, which was mostly CYP3A4 substrate and also time-dependent inhibitor and inducer. So as you can see, it's another example of similar compound. However, this compound was not as strong inhibitor. It was a little bit stronger inducer than inhibitor, but not as strong as when it comes to inhibition of CYP3A4 metabolism as the previous one. The compound was also inhibiting other enzymes in vitro. And the goal of the study was to predict drug-drug interactions with this compound as a victim when co-administered with moderate and strong CYP3A4 inhibitors, and as a perpetrator with a number of relevant probe substrates. So the model was done and the work was submitted to both agencies. The response from FDA was that model based on in vitro measured DDI parameters overpredicts compound exposure after repeated doses. Please improve your model and justify changes made in the model parameters. So in the first run, we used basically in vitro KIs and EC50s, all the DDI related parameters. And the model was reasonably capturing closely capturing day 1 administration, but was over-predicting the steady state dosing due to under-predicting induction effect, basically. And so we were asked to correct it. The Second point was that the food effect was not predicted correctly. And at the time when the prediction was carried on, there was no in vivo data. However, at the time of submission, the food effect study was already conducted and this data were available. So the agency asked to improve the PBPK model in terms of food effect and justify the changes made to the model parameters. And also they asked to predict additional DDIs with strong CYP3A4 inhibitors such as itraconazole and ketoconazole under fed condition after this model was corrected. The response from EMA was similar in some ways. They also complained about the time that the time dependency of induction is not well captured and ask us to improve this prediction. They pointed out to number of parameters, especially those that were fitted model parameters, to be explored with sensitivity analysis. And they asked also for qualification for two standard strong inhibitors used for DDI prediction, which were not included in the initial submission, which we provided. So agencies, in the end, agencies accepted additional information and analysis, and no additional clinical DDI studies were conducted. I have to say that during the process of this study, there were two clinical DDI studies conducted, which were part of the initial plan, since the compound was auto-inducer and auto-inhibitors, and when it comes to similar interaction with similar compounds, agencies were not certain that the model would predict the outcome. So these in vivo studies were used as additional validation of the predictive power of model, and in vitro data that were eventually provided, measured for these DDI interactions. Now I will move to the literature reported examples. Based on the information published last year in Clinical Pharmacology and Therapeutics, which lists a number of examples related to different type of submissions to both FDA and EMA, I picked some of the examples that are more interesting. One of them is related to transporter DDIs. It's trametinib, which is a weak BCRP inhibitor, and based on the in vitro data, this process was flagged as potential risk of in vivo DDI according to EMA regulatory guidelines. So since the sponsor already had GastroPlus model built for this compound, they predicted intestinal concentrations, and they predicted that complete inhibition could occur for the first 40 minutes post-dose, and partial inhibition of BCRP was predicted up to 1.6 hours post-dose, which was pretty much restricted to duodenum and jejunum. So after this analysis, recommendation was to limit the co-administration of a sensitive BCRP substrate to 2 hours post-trametinib administration. These data were submitted to the regulatory agency, which was EMA. The agency requested to further discuss the interaction potential between trametinib and drugs mainly absorbed from duodenum and jejunum, which they did by researching the University of Washington database, where they found the list of BCRP substrate absorbed within the 1 to 2 hours after oral administration. And they refined this list further to focus basically on the compound that may be potentially affected only by BCRP inhibition. This was submitted to EMA and accepted. The other two examples are related to the intestinal mechanistic modeling of absorption. The first one is for lesinurad, where model was used for dissolution specification. In this case, the IV and PO data were used for model validation, and they also had clinical data from batch that was bioequivalent to prove that the model was correctly capturing the in vitro to in vivo extrapolation when it comes to the particle size and in vitro dissolution data. And this is sometimes quite important point to have the negative control batch, which sometimes clients do not have, since especially when the compound is highly soluble. So this was this submission was accepted by FDA, was not submitted to the EMA. The second example is canagliflozin. Here, the sponsor basically changed the manufacturing process, which resulted in quite different particle size. And they used the model built on the initial original particle size and some non-clinical bioavailability study in big dogs to run sensitivity analysis for particle size to determine what range of particle size would produce in vivo outcome that would be still bioequivalent to the initial formulation, initial particles. The FDA accepted the study and requested some additional information, which were pretty much typical information that are requested by the agency when it comes to PBPK submissions. I couldn't resist but to show this cartoon because in the past the FDA has had a reputation of being overly demanding when it comes to drug approval process. But based on our experience, it has come a long way in recent years, and it has become more progressive and accepting of modeling results than the other regulatory agencies. And I have attended the FDA workshop called Flight Simulator, which has been related to developing complex generic drug products, and which was provided during organized during the last APS. And what I learned is that the FDA is open to proposal of using modeling approaches to establish bioequivalence for the test products, as long as these proposals include information about the modeling approach, scientific justification of the proposed approach, and in the end, model verification. They also encourage companies to discuss proposed bioequivalence modeling approach through the pre-ANDA development meeting. And I would like to thank you for your attention and ask if you have any questions.
Thank you very much, Grace, for the great overview, great presentation. To the attendees online, we now open up for questions. I already saw some questions came up. As mentioned earlier, you can write up your questions and directly using the questions pane on your control panel. We have a specific question here, Grace. For the first example, in response to the EMA's request for an in vivo study with strong COP3A inhibitor, will the study be conducted with single dose or multiple doses of the substrate drug that is with auto inhibition?
You know, it was quite a time ago, but I believe it was multiple dosing. In this case, when the compound is strong inhibitor of its own metabolism, so to to capture both processes, auto inhibition and inhibition, I would think that they would ask to run it for the number of days, but I'm not, we were not part of this later development, so I really don't know for sure what was asked. We can possibly reach out to the client and ask for the specifics later on and provide the answer via email.
Thank you. The next question, it seems like a little more general. How to develop and validate a model for bilayer tablet formulation? First layer IR and the second layer CR, control release.
How to develop and validate the model, right? So obviously the model validation would have to be done first with the IV and IR data. Typically if the IV data is not available, then the least formulated IR data, such as solution data, would be the minimum required to sort of separate systemic PK from the absorption processes. And so once the PK is validated with IR data, then the PK is the same for the between the immediate release and extended release products. The difference with the outcome or plasma profile for extended release products is related to the controlled release process, right? So the release rate is different and controlled typically by the matrix of the formulation. So in this case, one would have to first develop some in vitro, in vivo correlation for the extended release formulation, having maybe some pilot study. And then based on this correlation, if the formulation would be of that same type when it comes to control release, but release rate would be changed via amount of polymer in the matrix, the GastroPlus can handle both immediate release and controlled release component at the same time. So if the in vivo release is provided to the software based on the IVR, then it can be easily used. This kind of model can be easily used for guiding a formulation development of that sort.
Okay, thank you. The next question, how can you predict interactions that involve EHC, that, for example, a console with a statin?
I'm not sure if I got the first compound, but I understand.
Yeah, it's CONZOLE. Not sure it's a console or console with a statin, STATIN.
Statin, yeah. So basically the question is of predicting drug-drug interactions with statins, right?
That involve intrahepatic circulation.
Circulation. So if the enterohepatic recirculation is accounted for in the model, the model will use it for prediction of drug-drug interaction. The challenge is typically that statins are, some of them at least, are substrate for both metabolism and transporters. So I wouldn't think that enterohepatic recirculation by itself is a problem, but accounting properly for transporter interaction might be a problem, since the in vitro data for transporters are still not easily transferable to in vivo data. So
okay, so I saw the further comment from him is the conazole. It's not yeah, it's a type conazole is a statin, yeah. And in his comments, the problem is that the intrahepatic circulation does not occur except when they are administered in combination.
In combination with
These
what?
two, conazole with statin, when they administer in combination, they observe the EHC. I believe that's what they meant.
I think that would be very strange situation when typically the drug, if the drug has tendency to undergo enterohepatic circulation, it should happen, with or without interaction. Maybe if the EHC is related to the specific enzyme or the transporter, right? So if the transporter is involved in the interaction and one of the compounds is excreted in the bile and then absorbed from the intestine, that could be maybe affected in terms of the amount of the compound excreted in the bile due to the interaction, but I'm not sure. I would have to see more specific information.
I think Freedom is the person who raised the question. He said yes, and the conazole impacts cholestasis, and the statin is gluconidated.
Glucuronidated, yes. Okay.
Yeah,
so that could be potentially accounted for in the model if the process is captured well by model and basically interaction with the specific enzymes and transporters that are involved in this process would have to be captured well, but that would be very sort of, I would think, difficult model, first of all, to build, but not impossible.
Okay, we've got quite a lot of questions to address. Let's move on. The next question is the particle size distribution change case study as a result of a manufacturing plant move. Do you recall what was the BCS class of the drug?
I think it was BCS class 3. The compound was pretty much well soluble. The permeability was the major, I think, step limiting process. Yeah, I'm trying to find it. But yeah, there was BCS class 3 compound.
Thank you. And the next question, for development of generics, are you using PBPK module, especially in justifying dissolution specification failure or widening of dissol specs? Are you using mechanistic absorption modeling for genetics?
So yes, generics. In gastro, mechanistic absorption is always used because we use the ACAT for processing basically compound going through the given orally. So mechanistic absorption is always used. When it comes to PK modeling, gastro gives two options, either using compartmental model or PBPK model. And typically, from what we see, companies go for the compartmental model if it can provide sufficient description of PK. However, sometimes going to PBPK model is necessary. For example, you couldn't probably use the compartmental model for modeling statins or sort of compounds where you have to have transport inside the body, not only in the gut, but in other tissues, such as liver or kidney included in the model. So, and sometimes where also there is no IV data available and no simple IR formulation data, the PBPK is the only way to actually predict volume distribution of the compound correctly, relying on some prior animal studies where IV data were available. So it all depends on the compound and available data.
And he has a follow-up question related to this one. What's your experience with the FDA in the acceptance of such approach for generics?
Well, we have, as I said, very good experience. The typical, the shortcomings are typically with the proper validation of the model. We sometimes have clients coming back with the negative response from the agency, and typical problem that we see was that the model was not validated properly or was not used properly to really show that the compound would be still bioequivalent. So and sometimes the issue is with the lack of kind of negative control, as I pointed here with lesinurad case, that it was I would encourage everybody to read this publication by Pepin. It's very well written publication and really shows nice work done for this compound, and the model that was submitted, the data, and that really shows how to do properly submission. So it's all about how the modeling was done.
Thank you. Another relevant question along this one. Is it a good idea to provide a bioequivalence of BCS2 generic drugs to the reference on the market using in vitro dissolution together with simulation software? I think the answer is yes, right?
I guess. I'm not sure is it a good idea to provide bioavailability?
Bioequivalence, BE. Of the generic BCS2 generic drugs to the reference on the market using the in vitro dissolution together with the simulation software.
Yeah, I guess. It all depends in the context of what that would be used for, right? But if I'm not sure if I understand this question correctly, but if the intent is to produce new formulation that should be generic formulation, let's say, that would be bioequivalent to the existing market formulation, proving that this formulation is bioequivalent is a must. So.
Okay. Thank you. The next question, based on your experience, what is the most different point between FDA and the EMA in terms of regulatory acceptance of a PBPK modeling and simulation?
So, as I mentioned in my presentation, FDA, I think, is more progressive and accepting and less constrictive agency. EMA, since EMA doesn't accept the model files and cannot run the model, they typically ask for exhaustive report and also lots of validation for the models, other models that were used in the submissions, especially when it comes to drug-drug interaction predictions. Otherwise, typically, we've seen that the same work was submitted to both agencies, that by just EMA wanted a little bit more validation work when it comes to other compounds used, as I said, like in DDI predictions.
Okay, thank you. What is the scope of the approvals by the FDA or the EMA other than PBPK model simulation?
I think it was shown in this table, so I would encourage everyone interested to read this recent publication in Journal of Pharmaceutical Sciences that was just published, showing this is the number of submissions to Office of Clinical Pharmacology, which shows different type of submissions. So in this office, the DDI-based SIP DDI-based models are the mostly accepted. They do also accept other models for other indications, but I don't know if the number is basically related to the acceptance rate or just the number of submissions that try to avoid clinical trials in this specific area and use modeling and simulation to prove that the clinical trial is not necessary.
Okay, thank you. One more question. We really need to move on to the next presentation. This question is regarding for BCS class 2 or 4 drugs that are PGP or BCRP inhibitors, there are quite a few false positive predicting using dose divided by 250 milliliter versus the IC50 ratio. What is your experience with regulatory agencies when using PBPK to simulate intrasite drug concentrations and to predict effect on PGP or BCRP substrate to justify in vivo significant DDI is unlikely? If regulatory agencies have concerns, what are those?
So typically, as you can see from this list, DDI PGP related to PGP transporter involvement, or the BCRP is not mentioned, but I have shown example and we've been involved in study of that sort. If the model is convincing to the regulatory agency and shows that in vivo PGP effect or BCRP effect wouldn't be that significant, or how to, if it is of concern, how to avoid it by via shifting dosing of the compounds that could be affected, as one of these examples have shown. It is second highest accepted group of DDIs when you look at this table. So it sometimes depends also if it's easy to identify separate the PGP or BCRP effect from the other, let's say, transporters or transporters that could be involved, or enzymes in the disposition of the compound. So if you have, let's say, compound which is substrate for PGP and CYP3A4, it should be easy to account for the PGP effect, and I wouldn't see much problem if the model is built correctly to use it for drug-drug interaction predictions.
All right, let's get one more question. We'll have to hold. There are a few more. We probably wait until the end of second speaker presentation. Then we still have time for questions. So any approvals for protein molecules using PBPK?
So not from our side, that I know of. You can see that there were 2 submissions total for biologics. So but I don't know the details. So yeah, I really cannot comment on that.
Okay, thank you, Grace. Since we have another presentations and I was watching the time, so we need to move on to have Dabin to give us the case study. Then we will have time at the end to ask questions. I believe Grace will still be here and to answer.
Yes, I will be there.
Thank you, Dabin.
Can you hear me?
Yes.
Okay. So good morning, everybody. I think we just had a very nice talk from Grace, and so my topic is just on the particle size. So we will talk about use industrial case to show how did we justify the particle size specification using the biopharmaceutical simulation. It's not moving. So as we know that the API particle size is a very important property of the drug substance. It can affect different aspects of the drug product, like say dissolution rate, bioavailability, content uniformity, stability, and also will affect the manufacturing aspect, also like the powder flow, compactability. So in the SAH Q6A guideline, there's a very good decision tree on how do we set the particle size. So basically, you have to look at how the different if you have show the authority that if you have a tablet with different particle size, how would that impact dissolution, especially where that pass your dissolution spec, and how would that impact your bioavailability, and then also content uniformity, processability, those different aspects. So here we would just use a case to show how did we justify the particle spec using the simulation. We have some dissolution data, but not a lot. So the biopharmaceutical simulation, mostly we're trying to simulate how the drug actually get absorbed along in the human GI tract. But the human GI tract is very complicated organ, if not the most complicated. But it's very long and has very different hydrodynamics and different pH and different like volume, and also the drug once get mixed in the stomach and then the plug flows along the small intestine. And also there's a pockets of the intestinal liquid in your intestine, but it's not continuous kind of distribution. And also they have relative low solubilization power of the in the fluid because of the mostly is the low concentration of the bile salt and some lecithin to mesolyze your poorly soluble API, if that's your case. And also the absorption is facilitated by the sink condition created by the absorption. So the dissolved drug then get removed from the vicinity of your API and to facilitate the further dissolution. So in GastroPlus, use this so-called ACAT model, they divide the stomach, the GI tract into different compartments, like one compartment store for the stomach, and one for the duodenum, and several for the jejunum, ileum, and colon. And they set different volume, surface area, permeability, pH, and the transit time to different compartment, and also API divided into the three species, like the unreleased drug, unreleased drug, and undissolved drug, and the dissolved drug. Only dissolved drug can be able to be absorbed. So they mostly use the first order kinetics to describe the kinetics. So this is quite a useful tool because then you can use you can for your drug product, you can for the absorption process, you can dissect the overall process into disintegration or drug release for control release, or solubility, absorption, distribution, clearance, and before it go manifest as the PK profile in your systematic circulation. So you can assign different kinetics to a different process, and then you kind of build up a semi-quantitative kind of a math equation into this overall process to kind of assess how the individual process impacted the overall kind of effect. But sometimes it's not that straightforward, because a change in one place may affect multiple aspects in different directions. Like, say, for example, the food effect may be very complicated because a food can increase the viscosity, then it's gonna impact your disintegration or diffusion, and also change your solubility, and also change your gastric emptying pattern, and also in some cases it may even change your PK. Then it's very kind of the parameter, the simulation input gonna be very kind of tricky to set. For this case, this is a verapine. The verapine compound is is the first is PCS class 2 compound. Is a weak base, a relatively small molecule. And the IR tablet formation was approved in 1996 and is listed as an essential medicine for HIV/AIDS by WHO, and on the market also available as the extended release tablet. So this talk is mostly talk about how do we set up the particle size spec for the extended release formulation. So for the background for this IR tablet, we actually we have a particle size spec for the IR tablet, actually, which was systematically studied, and we manufacture different tablet with different particle size spec, different particle size from large to small, and we found the dissolution was very sensitive to the particle size, and we set the particle spec based on the API size and also whether or not they pass the QC dissolution testing. And we also has a bees study in human to confirm the adequacy of our particle spec for the IR tablet. So our tablet, the particle size spec is determined by the sieve analysis, and is set as a no more than 7%, greater than 250 micron, and low less than 80%, less than 125 micron passing the sieve. So for extended release tablet, with a different, it's very different situation, because the dose was increased to 400 mg, and then the release, of course, the release pattern is very different. Now it's kind of a matrix tablet with a lot of HPMC polymer in there. So first the polymer will swell, and then slowly the tablet will get eroded during dissolution or during the travel along the GI tract, and then the API particle get released and absorbed. And also we want to have this same particle size spec as the IR, so that will save us a lot of logistic issue, and also reduce the overall cost for the development. And we luckily we have IVC established for ER tablet with different polymer levels, but with just one particle size characteristic. And we also did a lot of mechanistic study on the regional permeability in human, and we have a good handle on the, like, say, what's the permeability in colon. So the overall objective is trying to first understand the effect of the large particle size on the bioavailability of the ER tablet, and want to demonstrate that this current IR API spec, particle spec, is valid for the ER tablet. And we want to accelerate the timeline by avoid manufacturing and testing of the additional large particle tablets, because there's later on we don't have like a API spec with large particle size. Then if we want to do it, we have to have additional manufacturing and or the tablet and the drug substance. So our overall strategy is we identify the key simulation input, and we want to support each input with multiple independent data sets, because we know that all models are wrong, but some are useful. So we want to show that our model, although it cannot really, the ACAT model is not a real GI tract, but we want to show that this kind of is still very convincing. We have confidence in the data and in the model. So first we look at the PK parameters, and then actually this is a bottom-up approach. We first look at the PK parameters, and then we look at the regional permeability, especially in the upper and the lower GI tract. And then we go a little further to set the parameter for the in vivo dissolution and the precipitation of API particle. And then we try to understand in vivo particle release and the type of erosion rate for the ER tablet. So we're trying to support each kind of aspect of the model with the independent data set. Then I think only after that we can convince ourselves in the companies that the model is robust and reliable, and then we can convince the reviewers and authority so that we understand how the particle size impact the bioavailability, and this particle size should be valid. So first, look at the PK parameters. This is we use the compartmental model. It's not a PBPK because this is relatively simple. We use one set of the two compartment model to describe. Actually, we can describe that the IV data, also PO oral data at 50 mg and 200 mg, and we at the 400 mg PO, like IR tablet, actually the clearance was showing a little bit different behavior, and but I think that at this even at the 200 mg PO like IR tablet, the plasma concentration actually is much wider than what we will be seeing for the extended release tablet. So we feel this at least within for the ER tablet, the PK parameter is linear, and it shouldn't change, or if we change the particle size. So second is we want to see what's the effective permeability in human. So first we test for the solution formulation, the Cmax would be proportional to the effective permeability. If you do the parameter sensitivity test, you see that if you for the fixed dose, if you increase the permeability, the Cmax gonna increase proportionally. And but this has a lot of caveats in there, because first, we don't have really we didn't have data to really solution formulation dosed into human, but we found that because we have this IR 50 mg, which should behave the same, very similar to that of solution, because the solubility in the stomach is very high, and this dose is relatively low. So this should also behave the same as solution. Second, in other cases, we see the double peaking in the PK, which is maybe explained by the gastric emptying pattern, because if the drug is not emptied in the duodenum at once, then this kind of a simulation, this assumption gonna fall apart. So we also see that about when we at 200 mg or 400 mg, there are a lot of double peaking in the PK, but at 50 it's not significant. So we feel confident that this is valid to kind of deconvolute the effective permeability in the small intestine using the clinical data. And also we did just calculate the in silico prediction based on the particle, polar surface area, the diffusion coefficient, the partition coefficient, and also the hydrogen bonding donor numbers. As those two pieces of information come together, we feel confident that the effective permeability in the upper GI tract can be estimated to be 4 × 10⁻⁴ centimeters per second. So next is trying to look at the permeability in the lower GI, especially the colon area, because that will be especially important for the controlled extended release, because the majority of the drug actually would be released at this colon area. But that information cannot be deduced from your IR formulation, because for the instant release, most of the drug absorption would be complete in the small intestine, and then you would have no information from those IR type human data. So here we use a very fancy technique. They can dose the API into different regions of the GI tract, and then we dose the suspension into say jejunum, ileum, and the different part of the colon. and we look at the Cmax and AUC, found that even if those at the colon, the Cmax and AUC is really kind of comparable to that in ileum and jejunum. So we feel this permeability or absorption rate in the colon and lower GI tract is gonna be comparable to to that in the upper GI tract. And later we also confirmed that allergy by looking at the real human plasma concentration profile for the controlled release tablet. You can see that even in this region, after like 4 hours of the oral administration, the absorption rate is really going on very still quickly. So which means that because after 4 hours, the tablet is most likely already in the colon, and then the absorption still going on very strongly. So that's kind of confirm our previous assumption. So then we kind of we settle on this PK parameters and also the regional permeability, which is the very important aspect of this model. And then we move on to this in vivo dissolution and the precipitation of the API particles. Like say, if you have just the API particle without the polymer from the ER, then how would they perform? So we use this modified Johnson equation, which is derived from the Noyce-Whitney equation. It's very generic and very well accepted model, which use the diffusion layer thickness model, where they assume that the thickness of diffusion layer is to be a 30 micron when the particle size is large, and when it's small, then the diffusion layer thickness is going to be equal to that of the radius of the particle. So we use the experimental data like determined by in the SIF or SGF. So uses the experimental measure data to just as input and to look at how the model how the prediction agree with the experiment clinical data. We found that it is quite good agreement, especially in the fast increase in the initial phase. But there's some kind of a slight difference between this region, because this we suspect it is mostly because of the gastric emptying pattern. Because for the 200 mg, we see a lot of double peaking, that maybe some of the drug is not the gastric emptying pattern we know is usually is erratic in human. Some is not a little portion of your gastric content gonna be emptied in your into the duodenum, that manifests as the small additional peaks especially after the lunch, so that you can see that can be is kind of a very common. So, and also we didn't include that 400 mg dose IR because that was we know the PK was a little bit, the clearance rate was a little bit different due to that high plasma concentration. So so now we have this the in vivo dissolution of the individual particles. Then I will go to the last piece of our model, which is how the extended release formulation actually release how fast they release the API particles in along the GI tract. So this is our formulation is a relatively simple formulation, so where the API crystalline particles is kind of embedded into the polymer matrix, and then polymer slowly swell and get eroded in the GI fluid, and then the individual particle be released and to then dissolve and then get absorbed into the systematic circulation. So we use this CRU module in the GastroPlus, which treat the tablet as integral tablet, like an intact tablet moving along the GI tract. And we use we use this our in vitro release rate as a starting point for our subsequent optimization. We fixing all the other physicochemical properties, physiological properties, and PK, all those fixed, and we do optimize how at what time point how much drug release would be able to fit the clinical PK profile. So this is so this left figure is how this the release rate of the particles as a function of time along the in the human GI tract, and the right figure is show the simulation and the actual data. You can see the first beginning there's a relatively fast release, maybe in the stomach, maybe due to the burst effect or some maybe high strong shear force in the stomach, and then there's a release is more or less linear up to like until the colon. And actually that's kind of agree with our kind of very linear release in vitro testing. So then the question, the assumption is for our future simulation is that this in vivo release rate is gonna be the same if you change the particle size a little bit, like make it larger or smaller. So that's I think that's the biggest assumption. So that whether that assumption is valid or not is very important. We have to really look into this and make sure that it will convince ourselves before we can convince other people. So first, we don't have large particles, but we do have some kind of a jet milled API, which has a much smaller particle size. You can see this is the unmilled material, assisted material, and after we're milling, the particle size is much smaller. So we made that into this matrix tablet, and we did the in vitro testing. So we can see that the jet milled material did show a little bit higher dissolution, like release, but overall this release rate is not really affected a lot. And the second piece of information we get from the IVIVC. In the IVIVC study, we didn't kind of vary the particle size, but we did vary the polymer level in the tablet. So we do have like 20% polymer, 35, 30, 40, and which has different in vitro release rate. And when we dose this tablet into the human, we found that actually they're within certain, like actually their in vivo study, especially between 20 and 30, this fraction absorbed, and the Cmax doesn't really change much. And our final concentration is about 25. So we feel that even if we have a slightly different the in vitro dissolution rate and also slightly different HPMC level, this in vivo dissolution kind of rate is not that variable in humans. So that gave us a lot of confidence in our future the accuracy of our future prediction. So once we have this, like all the pieces of our modeling is kind of in good shape, we hope that we feel confident. Then we go on to evaluate how the particle size impacts your bioavailability. Especially, we want to understand how, like, if you have a borderline particle size, how would that change your bioavailability? If you have a like small excursion, how would that translate to your change in the bioavailability? So first, our current particle size spec is defined as like no more than 7% greater than 250 and no less than 80% less than 250 or 125. So it doesn't really give you a real particle size as a simulation input for the modeling. So we first look at assuming a log normal distribution. If you change different particles, if you have like different particle size, then how much would be like say a greater than 250, and how much would be less than 125. So if you have a larger particle size, then you will be more particles greater than 250 and less particles like less than 125. Then we use the Excel's the most important tool, like use log normal distribution function in Excel. We look at it with the different, like say, distribution, the width of distribution, like D50 over D90 over D50, like 3, 4, or 5. And then different, if you have different particle size, how much would be kind of greater than the, how would that compare with our particle spec? So we located that if you have like 32 micron, then we'll have like say right at 7% above 125, and if you have 60 micron, then you have like represent as a small kind of excursion. And we want to use this like widest, kind of relatively high variability in the particle size with widest distribution width as our simulation input. So this is to show that our actual particle size used in the API in this manufacturing, and compare with our this blue line, the spec of particle size, and this red is excursion. Some represent some excursion. So that our API actually is well controlled within this spec, all the historic batches used for the clinical batch. So we then use this particle size kind of parameter sensitivity test analysis at 400 mg ER. So we look at how the particle size impact your bioavailability. So basically at the current particle size, we see like about 70% kind of relative BA relative to the IR. So you have a borderline with the simulation show that 69% are going to be your maybe most like the BA. But if you have excursion, like you may go down to 64. So so considering relatively high CV in the clinical profile, usually it's like greater than 30%, and so this even a small excursion in the particle size spec will still be bioequivalent to today's current particle size. And this just shows how the even at this excursion, this only slightly Cmax gonna be a little bit lower, but still the overall AUC is not that different. So we feel confident this is okay. So we include this simulation to the FDA as our justification for the particle size, and actually we didn't get any review question, and it's accepted, and now the ER is on the market. So this brings me to the conclusion. This field that GastroPlus model we can use that as serve as a knowledge platform to integrate, or like integrating existing in vitro and in vivo data. So that is a very good tool. And but during this model build, we need to really examine those key assumptions, especially the original permeability, the particle release rate, and then trying to confirm those with independent data set, so that we can convince ourselves and also the future reviewer. So this data is very very good data, and we showed we convinced the reviewer that this would have minimal impact. And we actually, the justification was sent without additional tablet manufacturing and the dissolution data, and we accepted without further questions. So we feel this systematic and semi-validated modeling actually is very powerful and effective to reduce the time and cost of the drug development, and it's a great we can use this model to generate new knowledge based on our existing data. So I want to acknowledge Todd Darrington, which he was really bring this question to me, and we have a good discussion and figure out and trying to fish all the useful information from the very big haystack of the existing data, and trying to find the pieces together, put pieces together. I also thank Jane as the mentor and the manager at that time, and also Charlene to give this opportunity to present, and also Vijay as a good colleague at that time, and we had a lot of interesting discussion. I think that's all my presentation, and can may ask any questions. Thank you.
Thank you, Dabing. Thank you very much for the very great presentation. At this point, we will open up for questions for both presentations. As questions probably are coming up, and we have still a few questions for Grace that have not been answered. Maybe while we're waiting for questions for Dabing, we can still complete the answers for the question for you. One question related to your talk is, what is your experience in creating model for any oral dosage forms having very high intra-subject variability? How reliable is the predicted PK parameters for such high variable drugs?
So we obviously deal with this kind of compounds, and the way we try to deal with it is to find out the reason for this high variability. Many times we group the subjects based on the similarities of plasma concentration time profiles and try to model not every single subject but just the group average of the group, and find out the reason, and usually everything falls in place after this kind of exercise is done. In the end, to use this model in the future, population simulations would have to be used with the variabilities observed in the individual subjects would have to be accounted for in this population simulation. So typically either the group of subjects or individual subjects would have to be modeled in order to find the coefficients of variations for parameters responsible for variability.
Okay, all right, thank you. The next question for population PK of IR formulation, why Simulation Plus recommends .crd file instead of .dsd file?
For population simulations?
Population PK of IR formulation.
For IR formulation? Technically, IR formulation shouldn't use control release profile, which CRD stands for. IR formulation should be, if it's a simple IR, I would say, should be accounted for with particle size data used as the input to calculate in vivo dissolution. So I am not sure what we would have to have probably offline talk with this person what was specific case and why the control release input was used for modeling immediate release formulation. Right,
this probably requires some follow-up and to clarify the question. The next question is, do you have any experience of PMDA submission? I believe this is a
Japanese agency, medical devices.
Case.
No, we don't have direct experience. We've done number of projects for Japanese customers, but we never heard back how these models were used, whether any of this work was used for the submissions to the regulatory agencies.
Okay. The next question is, any para-IV approvals?
Actually, what is para-IV? I don't know.
That's what I'm PARA, para-IV approvals. See if, I think the individual has left.
Yeah, okay. We would have to have I don't understand what is para-IV, so we would have to have some clarification.
Right. Okay. I think that's probably all for Grace's talk, and then the question for. All right.
I have just one more, if I can. There was a question previously about the BCS class of the drug for which particle size was changed during the manufacturing process. I checked, it was actually BCS class 4, formally BCS class 4 compound.
The example. Oh, someone actually from audience answered the question. It's not a para IV, I'm sorry, it's para 4 approval. What is the para 4 approval?
Yes, well, I'm not sure what is para 4. I'm not. It's the kind of.
Anyone from audience could.
Yeah, maybe if they can, somebody can shed the light what it stands for. Para IV. I'm trying to Google it.
Patent non-infringing approach. I still do not understand, but I have there's a question for Dabing. Dabing, how did you decide on the numbers of bins of PSD, the product size distribution, to put in the software?
Yeah, for me, since we use the min and the SD, so I just use the highest bin available. I believe it's 15 or 10. I forgot. Yeah.
Okay. All right. Let's see. Any more questions? I think we gone through all the questions except that this para 4. Do not really quite understand. Okay, Tycho has a question. Does BI have experience with, the question for Dabing, have experience with successful absorption model without IV? Because in your modeling, we do have an IV, to validate the model first.
Yeah, I believe because we don't really have a real case, but in some internal other compound, we do just show that this PK compartment of PK parameters can describe multiple oral PK profile and show that linearity, and then I believe that should be okay. But I cannot say say for sure.
Yeah, for internal development, I can comment a little bit for to support the development, the formulation product development. So if oftentimes IV lag is not available in the clinic, especially early stage, we have used the solution formulations and also at the low dose, sometimes to assume that the drug will be close to completely absorption, and to, as Dabing mentioned, to validate the model by using multiple different doses, and that's one approach we have been used. But I don't think we have experience to actually submit this type of the approach to the regulatory approval in any of the submissions. Okay, the next question is, I'm surprised that the Tmax for the ER was around 24 hours based on the length of the GIT, the gastric intestinal tract. Question for Dabing.
Yeah. So I think this is it's an interesting case because that's the data we also kind of feel surprised because usually the Tmax should be around like 4 or 5 hours, like where the major absorption site would be in the small intestine. But for this one, I suspect that maybe this compound is relatively small and well absorbed even in the colon. And maybe that's and also combined with a relatively slow clearance rate, and that maybe even that suggests that the colon is also a good absorption site for this compound.
Maybe if I can have one comment, was the controlled release dispersed type or integral tablet type?
It's I think it's maybe a combination. It's not a full kind of intact tablet, but still maybe some disintegration maybe occurred. Different fragmentation also occurred.
Because sometimes when the control release formulations are of the integral tablet type, it means they don't disintegrate, and their size exceeds the size of the stomach pylorus. And this kind of formulations can reside for extended period of times in the stomach. And in these cases, the very long Tmax can be observed too. So yeah, there might be multiple reasons for that.
Yeah, let me see if there are any people raising their hand when they are calling on the phone here. Let's see. All right. Mala Durai, are you still online? I saw you raise your hand. No. All right. I think we are getting very close to the end of the webinar time, and thank you both, Grace and Dabing, for the very good presentation. And in case, for the audience, in case you have additional questions after the webinar, please, you can go to our GastroPlus user group LinkedIn website where you can post your questions. So your questions will be addressed by our members or members in the Simulations Plus. So thank you again, Grace and Dabing, for your presentation and answering all the questions. And for the audience, thank you all for your participation. So you may now disconnect from the webinar. Thank you, everyone.
Thank you.
Thank you.